website under the Evaluation tab or by following the link in the chat box. Those of you who are online, please enter the full name and credentials of everyone in attendance in the chat box at this time so that we may keep attendance. Your comments are very important to our planning committee. Please turn in the comments at the collection box at the entrance. As you know, all sponsors of CME are required to execute a conflict of interest policy, and speakers have to tell us about real or apparent conflicts of interest. Dr. Jesse Owens, Kalpana Kalyanpur, and Kaz Nakagawa have disclosed that they have no relationships with commercial supporters. Dr. Brad Wilcox and Dr. Rich Alsop are consultants for Cardax. So next I'll introduce our speakers. We're going to be having 5 speakers today, so each one will be speaking for about 10 minutes. I will ask the audience to please hold your questions until the very end, and then we'll have 10 minutes at the end for all questions. The Center for Biomedical Research Excellence, or COBRE, is funded by the National Institutes of General Medicine Sciences that supports multidisciplinary centers that strengthen institutional biomedical research capabilities. The Kuakini Medical Center's COBRE has a research focus of longevity and was funded for 5 years starting in the fall of 2019. The COBRE team will give an overview of their research on a variety of topics. To start us off, I'm going to introduce the principal investigator of the COBRE, Dr. Bradley Wilcox.
Hi. I'm pleased and honored to be here today. I'm going to check to see if the microphones work here. Test. Oh yeah, here we go. Okay. I don't like to be bound to the podium. I like to walk around. So I'm going to talk a little bit about this Center of Excellence on Aging. I'm really pleased and super excited about this center. It's a big honor to have a center like this. We've worked really hard at Kuakini, as you know, for a half century or more to build a data set and a biorepository and a clinical repository to really try to understand a lot of age-related diseases, and we're also focused on aging itself. So, anyone remember 1929? Come on. Okay, just kidding. We were born in the same year. And that wasn't 1929. So those are the early days. I'm sure you've seen this slide before, but since those days we've come a long way. Since 1965, we've had continuously funded research from the National Institutes of Health, which is a really, I think, amazing accomplishment. And we built this data set, and it really speaks to the foresight of the administration at Kuakini to really support this, because if it wasn't for the administration supporting research at Kuakini, we wouldn't have the center, we wouldn't have these data sets. So as you all are very aware of, the Kuakini Honolulu Heart Program was the first major study at Kuakini. It was part of the Nihon San study that compared Japanese in Japan, in Hawaii, and San Francisco in terms of disease epidemiology, mainly cardiovascular disease. It led to many other outcomes, including cancer studies. And then in the 90s, we added aging studies. And then in the 2000s, we added genetic studies of lifespan, as well as health span. And now we've recently begun an offspring study. So this is the current status right now of the Honolulu Heart Program. I guess we're in year 55 now. We're going to have to update this slide. So we're continuing to track mortality and other outcomes. There's about 50 of the original 8,006 men still alive. The youngest one, I guess, as of January, will be 100. So that's the youngest one. The oldest one, currently I believe, is 106, but that's a moving target. We have many collaborations with including the medical school, national collaborations, international collaborations, and we continue to expand our collaborations. One of the major strengths of the Kuakini Research Program is the specimen biorepository, which has close to If you add them all up, close to 500,000 biological samples that have been stored. We have samples as far back, we have any from 1965, come on? Okay, the 70s, the early 70s. So it's really, for aging research, that's what you need. You need a longitudinal data set where you can look at how people aged over time, rather than just taking old people and comparing them to younger people, which is not the ideal way to do it, but that's how most aging studies are done. I think most of you know why the Honolulu Heart Program, Honolulu Aging Study are unique. I don't think I have to go into it in any major detail, but the study is very well known. It's got a wealth of data. We're getting into almost 900 publications, and we've started the next generation of studies. And this new grant, the Center for Biomedical Research and Excellence on Aging, is going to be funded for 5 years, as Kamal said, and this is how it looks. This is the organizational structure. So we've got the board of directors of Kuakini here, Mr. Kajilara, the president and CEO here, Mal here, me here, Dean Hedges here, and the directors of various in-gray and co-gray cores, including the biostatistics core, the Institute for Biogenesis Researchers collaborating with us, Steve Ward here today, and Dr. Gershenson, diabetes co-gray over there. Thank you very much, Dr. Gershenson. And we are also trying to establish collaborations with the microbiome program. So our focus is on healthy aging, which is really the intersection of avoiding disease, maintaining high cognitive and physical function with life, and maintaining an active engagement with life. So we received a really major boost in the mid-2000s. Actually, 2008, we published a paper that was the first paper that ever showed that a gene that was conserved across evolution in model organisms of aging was also something important human aging. This was the FOXO3 gene. It's a stress resistance gene. And so this whole program that we're building with this Biomedical Research Center of Excellence on Aging is to try to figure out how this gene works in a network and how these networks relate to other networks in terms of aging phenotypes. So you'll learn more about those phenotypes when you see the 4 major projects that we're working on, but that's the common theme. So this is how it works. We've got an administrative and mentoring core, and the clinical and translational core. We're enrolling 800 of the original Kuakini offspring, Kuakini Honolulu Heart Program offspring, 400 men, 400 women. We're going to be taking biological samples also from them, as well as doing a lot of clinical and functional aging-related tests. We hope to be able to follow them again in the future to establish a new longitudinal study. The projects, I'll just very briefly, we've got one from Jesse Owens, who's doing a mouse model for FOXO3 survival and health. Phil Davey, who's doing the effects of FOXO3 on stem cells and telomere. Kaliampur, who's doing the effects of FOXO3 on brain aging, and Kaznak Gawa, who's doing the effects of FOXO3 on on stroke, as modified also by ApoE. So they'll explain in more detail what they're doing, but they're all projects that have interrelationships and strengthen each other. We have some great collaborations with the University of Hawaii. Several of our new investigators came from the University of Hawaii. We've got mentors local, we've got mentors on the mainland, we've got international mentors, and we're working with the other 3 UH Cobras. Of course, none of this would be possible without the amazing team that we have, our clinical staff, recruiters. Some of them are in the audience here. And we really couldn't do this without them. And they've been with us, some of them almost 3 decades, right? So I'm going to move on from there. Did I make my time quota? Oh yeah, okay, it's good. So next up is Dr. Jesse Owens, who's going to talk about his mouse model and how he's going to cure aging.
Welcome to my talk. My title is Using Mouse Models to Study FOXO3's Role in Healthy Aging. So first I'm going to talk about what my lab used to be interested in. So basically what we, and we're still interested in, is basically what we do is try to develop tools to deliver a gene of interest, say a therapeutic gene, into the genome. And we want to do this at a specific address, a specific sequence. And the reason for that is if you can deliver a gene of interest to, let's say, a safe harbor, a sequence that's not near an important gene, you wouldn't disrupt cell function. Also, if that gene of interest were to fall inside of heterochromatin, your therapeutic gene might be turned off. So we're interested in taking that gene of interest and sending it to a specific place to make sure that we can do that safely and express our gene efficiently. So this kind of idea that I came up with was this idea of delivering a transposase to the genome to a specific sequence. So you might have heard of CRISPR technology. I'm going to briefly just kind of explain what my technology is, which is a little different. We borrow pieces from CRISPR's, and what we do is we make a guide RNA, which binds to a protein called Cas9. And that guide RNA is going to guide our, my animation's not working, but that's okay. So what it does is it guides the piggyback transposase to our guide recognition sequence and integrates nearby. So you can see the gene of interest is now located near the guide recognition sequence. And so what we're basically doing is we're localizing a transposase that usually integrates randomly to a specific sequence so that it integrates nearby them. And using this new technology that we just invented, we were able to identify insertions in the little black lines. This is a gene that we were trying to target next to our targets, which were these little red lines. And what we found was a bunch of insertions occurred nearby those sites, and so we were successful at hitting those sites in the genome. We could have gone to millions of other places, but we found that they landed kind of nearby. We used PCR to identify those targeted insertions, and that's the PCR products. And we used quantitative PCR to show that there was only one insertion in cells that we'd isolated. So we know that we've got one insertion, and we know where it went, and so we can be confident that there aren't any off-target insertions that might have disrupted some important gene. We also showed that the cells glow nice and bright after many weeks, indicating that the gene of interest isn't being turned off. So we just published this a few weeks ago, and this is the first demonstration that you can use RNA to target a transposon, which is the DNA, to a single user-defined genomic location. Now my next steps are going to be evolving these tools to be more specific and more efficient. So we're not there yet, but at around this time, I joined Brad and Rich, and now what my lab has done is kind of split off, and we've got kind of two interests that I'm hopefully going to get into are related at the end here. So now what we work on is FOXO3 and aging, in addition to the transposon stuff. So as Brad mentioned, FOXO is a pro-longevity gene. It's got a bunch of really interesting mechanisms for letting you live longer, making you live longer. It protects adult stem cells. It improves immune function. So when you have a dietary restriction, if you remove FOXO, dietary restriction doesn't work. So obviously, we've got something that's very important here. It has tumor suppressing activity. It improves autophagy, and one of the main ones is it protects against stressors, like Brad mentioned. So currently, they've only knocked out FOXO in mice. Nobody ever made more FOXO in mice, and so I thought this would be a nice idea. And our transposon system is really great at making mice. This is a mouse that we made with a really huge insert. You can see its unsuccessful litter mate is dark, and then this red here was the one that we made glow in my lab. So you can make full transgenic mice in a single step with this approach, and we published this. So the point is that transposons are really good at making mice. Now, why are we bringing in an extra copy of FOXO3? Well, we've got these kind of two haplotypes that Brad had mentioned, and what we think is happening is when you've got stress with the common haplotype, you get low FOXO3 protein induction. when you get stressed with the longevity-associated haplotype that we showed when you have that GG in humans, you live longer, we think that we get higher FOXO3 protein induction, and they've actually shown that in a paper. So what I was thinking was we could put in an extra copy of FOXO3, and now when we get that stress, we're going to get the high FOXO3 protein induction, kind of similar to the longevity haplotype. So we're trying to mimic that haplotype in the mice. And what we're going to do is we're going to express this from endogenous promoters. So what that means is we're going to use the natural enhancers and promoters to drive the gene. We're not going to overexpress it. And I think that's very important. And it's a lot more difficult to do this, but this is the kind of creative thing we came up with. So in a normal cell, you've got FOXO3 expressing cells, and you've got a lot of cells that actually don't express FOXO. It's not supposed to be there, right? And we think that when you've got stress, what happens is the FOXO3 production turns on in the FOXO3 expressing cell. No stress, no FOXO, and cells that don't express FOXO, no FOXO. This is what's supposed to happen most of the time. What a lot of labs do is they overexpress proteins of interest. That's like the common way to do it. If we were to do that with FOXO, we would have this situation where we would have FOXO turned on with the stress, which is the thing that we want, but then also without stress, we would have FOXO being turned on, and it might not be doing things that we want. And also, there's non-expressing cells that should never have FOXO, and they would also be expressing it. This is especially a problem for immune cells, and so we got to be careful to not express FOXO where it shouldn't be expressed. It might, like I said, it could be harmful to the cells if it's in the wrong, if it's expressed in the wrong area. So another thing is development is very complicated. FOXO is a very important gene. It's a transcription factor. It regulates many other genes. There's a good chance that if you were to express it, even that extra copy in the cells that are supposed to have it during development, it might mess up development, right? So what we've done is we've got it so that we turn on that extra copy whenever we want. We choose when that second copy turns on. So that we'll definitely wait for development to be over. The other thing we can do is we can wait for the mice to age to adulthood and then turn on that extra copy. And what would be really neat about that is, let's say you wait for a mouse to get to an adult, then turn it on, and then still see some sort of therapeutic effect. That would indicate that FOXO therapy would work in adults. You wouldn't have to take it from being a child, from the age of a child to adulthood. What we're hoping is that adults that have already aged could take a therapy and still get some sort of beneficial effect. And so that's why it's really nice to have this ability to turn it on later. So this is the kind of flow chart for my grant, and I'll just kind of quickly take you through it. So first we had to synthesize the FOXO gene, put it into a vector, and then generate mice. We just got our mouse 3 weeks ago, so it's glowing green, and with this mouse, if you induce the second copy, the mouse no longer turns green, and it now has a second copy in every single cell in its body. But again, it will still be under its own promoter, so it will not be active, it will not be driven unless it's supposed to be driven. So it's a pretty complicated little mouse. We're going to turn it on and off at different time points, and then we're going to take those mice and we're going to assay them for aging phenotypes. A whole bunch of different phenotypes that show whether or not these mice are aging healthily or not. We've included ROS for reactive oxygen species, stem cells in collaboration with Phil, and some other stuff like heart function and glucose tolerance. So our future goals, and I think this is my last slide, is this is kind of long-term stuff, but what I'm really hoping is that we do see, we're very hopeful that we're going to see that bringing in this extra FOXO is beneficial. It's been shown to be beneficial in other organisms, lower organisms. So what we hope to do eventually is use our same vector that we used to make the mice to deliver the gene to an adult mouse. So now we're not talking about an egg that develops into a mouse where that gene is in every cell of its body. We're going to take an adult mouse and then add it as if we would to a person, like an actual therapy. So systemic delivery of FOXO throughout the adult body could someday be possible. And if we use our targeting vector that I described earlier, and once I figure it out, get all my evolution done, that actually might be safe. And so ideally what we would do, maybe someday, would be take that extra copy of FOXO or something similar to that, deliver it to a specific sequence in the genome so it doesn't disrupt anything, so it's a safe therapy, and then hopefully get some mice to live a little bit longer. And I think that's everything. Thank you.
Morning. Hi, I'm Rich Ossoff. I'm actually the Deputy Director of the Code Grant. So I'm sort of the Mike Pence of the Code Grant. So Brad unfortunately passes away in the next few years. I can step in. Okay.
Lots of your longevity, I'm not going anywhere.
That's right. I'm more likely to pass away. Okay, so Phil's not here, so I'm filling in. I'm going to talk about his project, which I'm happy to do. Some great project. And let's see, winner? Yeah, so he's got a few aims. So his project will assess the impact of human longevity bearing the FOXO3 on telomeres and stem cells. So it's more of a sort of a biological study using samples from the HHP cohort. And that's essentially what it's supposed to do. The two possible mechanisms are telomeres of stem cells, and understand specific mechanisms of healthy aging which may identify targets down the road for interventional therapies, long-term. So, Brad already explained about FOXO variants and its improved health span longevity. But in the initial study that was done here about 12 years ago, was now replicated in over 12 different cohorts around the world. Talked a little bit about FOXO functions. Previously demonstrated correlation between telomere and FOXO genotypes and function of age. So this was published a couple of years ago. We'll talk a little bit about that as we go along. Background data. So this was a cross-sectional study. So what Phil wants to do in his first stage is actually do a longitudinal study with samples from the HHP cohort, which will be a much more powerful and compelling argument if it all works out that telomeres are related to the FOXO genotype. And then also look at what people haven't done yet in humans is association between the FOXO allele and stem cell frequency and function. So really cool stuff. As I mentioned, he's going to do a longitudinal analysis on the telomeres, cross-sectional analysis on the numbers of blood stem cells and neural stem cells as a function of FOXO3. So the telomere aim, some of the background data, as I mentioned, is shown here. So this is our preliminary study here done in Okinawa cohort, where you can see people that don't have a copy of the longevity allele, males and females, showing green and red, all show marked telomere attrition as a function of age. But if you have one or two copies of the longevity allele, shown by the dashed black and blue lines, there's very little telomere shortening. So this is a pretty good argument that there's not much telomere shortening going on if you have a protective copy of the FOXO3 gene, but if you lack one, then you see a fairly dramatic shortening. So this is something we're quite happy to see, and is what sort of drove us to say, well, what happens if you look at longitudinally, which is sort of a more robust analysis of the effect of potential effect of FOXO3 telomeres. So that's aim one. Aim two, this is all Phil's data, so he's got a lot of background on stem cell analyses, and so he's essentially going to look at stem cell numbers as a function of the FOXO3 genotype numbers and FOXO3 expressions relative to cells. So FOXO3 frequency and function in both hematopoietic and neural stem cells. I'm not going to go into the nitty-gritty details of this, but there's all kinds of secondary endpoint assays that he's going to look at. And some of the intended outcomes. So regarding the stem cells, actually, what Phil didn't mention is that FOXO3 actually does have a really important role in stem cells. Others people have shown that if you knock it out, specifically in hematopoietic stem cells in mice, you don't see an immediate phenotype. But once you transplant those stem cells from one mouse to another, you really see a very weakened ability of those cells to repopulate the immune system of the transplanted mouses compared to wall stem cells. So it's got a really important role in, at least in hematopoietic stem cells, likely other stem cells as well. And he'll be interacting with the other investigators on the cohort. Jesse looking at the mice and the telomeres and stem cells and the FOXO3 transgenic mice as well as Kalpana, and helping out the telomere analysis with respect to brain aging. So there's a lot of interaction going on with Phil's project and the other projects. So that's about it. I think for Phil's just final summary slide. And, top one.
So here's the title of my project. And this is a brain MRI study on the impact of FOXO3 on brain aging. And so we're looking specifically at changes in the brain that occur with aging. There will be 100 Kuakini HHP offspring, for instance. And the indices of brain aging that we use from MRI will be on brain structure and function, which provides some information about atrophy and functional disruption. So as we know, the protective variant of FOXO3 is associated with increased longevity. So this study hopes to find out how brain structure and function are associated with FOXO3 genotype and also with aging at the level of individual cells, which we'll assess using markers of biological aging in white blood cells. So normal aging is typically accompanied by gradual brain atrophy and cognitive decline. You see these three MRI scans at the top. The one on the left is a young adult, then comes a 94-year-old non-demented person, and then a 77-year-old person with dementia. I don't know if you can see from there, but we do note progressive atrophy, atrophy in the two scans on the right. And that's manifested by enlargement of the ventricles, that's the most obvious. But there's also widening and flattening of the cortex as the gray matter and white matter is lost. And the two plots on the bottom, the one on the left shows gray matter volume as a function of age, so there's a steady decline of gray matter as you get older. And on the right is white matter volume as a function of age. That loss is not as striking, but it does occur after middle age. And cognitive changes in healthy aging include slowing of processing speed and declines in executive functioning and episodic memory. There have been a lot of studies of gray matter in aging, atrophy in aging, and one of them is the UC Davis Aging Diversity Cohort. This is a longitudinal study of 460 very diverse older adults. And their cognitive spectrum ranged from normal cognition to dementia. And the study found that faster cognitive decline was strongly correlated with greater gray matter atrophy. So this and other studies tell us that it's very important to study the cortex of the brain, not just other regions. So we'll be doing this in the program. A quick review of telomeres. They're the protective caps at the ends of chromosomes, the DNA repeat structures that undergo shortening during mitosis. So every time the cell replicates, the telomeres shorten. They're important because as they shorten with replication, when the telomere length reaches a certain critical level, the cell becomes senescent or undergoes apoptosis or cell death. Aging is the result of a decreased number or function of cells, and longer telomeres are correlated with longer lifespan. So telomere length is like a biological clock that provides a marker of biological aging and susceptibility to disease. Shorter telomeres are associated with older age, but also many conditions, including the ones listed here. At the bottom are poor cognitive function, increased risk of dementia and death. Most or all of these adverse outcomes are also associated with inflammation and oxidative stress. So my project will include measures of those. And cell aging can be triggered not just by telomere shortening, but by telomere dysfunction. Telomerase is an enzyme that maintains the length of telomeres by adding DNA, nucleotides to the DNA. And telomerase activity can be inhibited by oxidative stress because telomeres are very sensitive to that. But there are studies that show that neurodegeneration has been associated with increased telomerase activity. So it's not always clear. We can't always predict what direction the telomerase activity is going to go when we're looking at brain aging or other conditions. The study participants will include two groups of interest. There are 50 carriers and 15 non-carriers of the protective FOXO3 polymorphism. The age range is 45 to 85. And this is a cross-sectional study, so each person is assessed once. They all have a brain MRI, structural and functional brain MRI, and cognitive testing. So the first two aims are going to examine brain. are examining brain structure in relation to FOXO3 and telomeres. Brain structure is reflected by regional brain volumes and measures of cortical thickness that we get from structural MRI. So we'll look at associations between those and FOXO3, as well as telomere length, telomere activity, and peripheral blood space. We expect that non-carriers of the protective FOXO3 allele will have lower volumes of brain white matter and thinner cortex, and that those deficits in brain white matter will correlate with shorter telomere length and changes in telomerase activity. To assess brain function, we are measuring something called resting state functional connectivity. This is where certain brain regions show synchronized activity in their spontaneous signals. And the brain regions can be far apart in the brain, but they're functionally related. And these regions that have synchronized activity are believed to represent resting state functional networks or intrinsic networks. These have been mapped extensively. This is one map of the networks, and here you can see that they include visual, language, attention, and so on. And these networks are active during rest, which means that the person in the MRI scanner isn't really given a task to do. The person's just thinking of anything. And they're also active when a cognitive task is being performed. What's relevant for this study is that resting state functional connectivity becomes weaker with aging. So we want to see how that is affected by FOXO3. So the aims 3 and 4 look at, first of all, resting state functional connectivity in relation to FOXO3 genotype and telomere length and telomerase activity. And then we're going to take all the brain integrity measures that we have, meaning the volumes, cortical thickness, and resting state functional connectivity, and FOXO3 genotype, and see how all of those are related to oxidative stress and inflammation, markers in the peripheral blood. And we hypothesize that the non-carriers of the protective FOXO3 allele will have weaker resting state connectivity that correlates with shorter telomere length and changes in telomerase activity. And they'll also have increased oxidative stress and inflammation, which will be associated with smaller brain volumes, thinner cortex, and which is lower resting state. So finally, once we've determined all these brain differences that are linked to FOXO3 genotype and telomeres, inflammation, oxidative stress, how does that all relate to cognition? So that'll be the last thing that we do. We will use the results from neurocognitive test performance and see what relationships are. To summarize, the study will provide insight into the impact of FOXO3 on the brain. We hope it'll clarify mechanisms of healthy aging, and perhaps it'll contribute to therapies that slow human aging and improve quality of life. So thank you, and I guess we're taking questions at the end. Is there any questions? Thanks.
Hi everyone. Kaznakal. I'm a neurointensivist neurologist at Queens. I'm a clinician. I just want to see, raise your hand if you're a clinician here. Yeah. So it's great. So it's great to see, it's a full spectrum of research coming from Jesse's very basic science. Mine's a lot more fun. So my project is really looking at the impact of both FOXO3 gene and also ApoE gene on the incidence of basically hemorrhagic stroke, kind of a remarkable over. Just to give you a basic sort of background of stroke, we have both ischemic and hemorrhagic stroke. So ischemic stroke is when you get a blockage of the blood vessels, and that part of the brain basically damages and dies. And hemorrhagic stroke is when the blood vessel ruptures and causes bleeding and causes mechanical damage to that area of the brain. Both hemorrhagic and ischemic strokes can cause damage and can be disabling. Just to note that this is the mortality, 30-day mortality after the first column is heart attack, ischemic stroke is the second column, the hemorrhagic stroke is the third column. You can see that the mortality rate is way higher than hemorrhagic stroke. It's very high mortality condition. And just to note that the blue column is all the comparable other countries, non-US countries, and green is us, US. So you can see that for a heart attack and ischemic stroke, we have a lot of tertiary intervention, like coronary stenting, TPA, thrombectomy, a lot of fancy acute intervention that really brings mortality down. But for hemorrhagic stroke, there's not much intervention you can provide acutely, so the mortality rate for the US is no different from other countries. And why is that relevant to our population is that unlike Caucasians where the hemorrhagic stroke accounts for only about 10% of the strokes and the rest are ischemic strokes, amongst the Asians, it's about 30% of all strokes, and so the burden from the hemorrhagic stroke is way higher amongst the Asians and Asian Americans. So what causes hemorrhagic strokes? About 70% of the cases due to high blood pressure. So you can see on the graph on the right that there's a linear correlation between your blood pressure, baseline blood pressure, and your risk of having a hemorrhagic stroke. Literally, every 10 points in systolic blood pressure almost doubles the risk of stroke. And the small vessel, we call it a weakening and microaneurysm formation eventually ruptures. It's unclear why some people get basically the long-standing high blood pressure, the blood vessel closes up and causes ischemic stroke, and why some people get a rupture and cause a bleeding. But there's I don't know why it says one, but it's supposed to be two. The second cause of a hemorrhagic stroke, which is amyloid angiopathy. And this happens amongst the elderly population, and it's nothing to do with the cardiovascular risk factors. So you can be, 80-year-old with no vascular risk factor and can have this hemorrhagic stroke. So what is an amyloid angiopathy? So this is a beta amyloid that you see amongst the Alzheimer's population, but this is a specific condition where that amyloid goes right into the blood vessels alone, and sometimes doesn't even go into the brain tissue. So you can have non-demented elderly person with a lot of amyloid deposition within the blood vessels in the brain. Or you can have Alzheimer's patients with a lot of amyloid in both the brain and the blood vessels. So obviously, if you have dementia, your likelihood of having this in the blood vessels is also high. So why does this amyloid, go only to the blood vessels or only to the brain tissue? So the precursor for the amyloid gets cleaved, and depending on where it gets cleaved, if it's between 39 and 40 residues, that amyloid tends to go into the blood vessels in the brain. If it gets cleaved between 42 and 43, then it tends to go into brain tissue. I'm not sure how that happens, but that's just the data. And when this amyloid gets deposited in the blood vessel, it causes a sort of splitting of the blood vessel wall, causes necrosis of the blood vessel, sort of tissue, and it causes microaneurysm and bleeding. So, we used to think it's this bad amyloid production that's the problem, but it turns out not to be the case. It's really production of the amyloid is staying amongst everyone. It's the clearance. It's the sort of like a gutter, and things have to get cleared. So amyloid has to be made and has to be cleared constantly. Then it's not going to be toxic to the brain itself and also to the blood vessel. So there's something about this sort of clearance that's causing people to have amyloid angiopathy. So what is this thing that's causing the clearance? It's the apolipoprotein B. So it's a lipoprotein, so it's a protein that binds to lipids to form the lipoprotein complex, and it helps to transport these lipids through the lymphatic and circulatory system. And it turns out that if you have a certain apolipoprotein B, then it clears the amyloid better than the other. So ApoE has E2, 3, and 4 alleles. ApoE3 is the majority of the case, 64% of the time. And if you have that, then it's fine. Like you're not at higher risk for amyloid angiopathy. But if you have ApoE2 or ApoE4, then you're at high risk of having cerebral amyloid angiopathy, that hemorrhagic stroke in the elderly. Really, there's only like one observational longitudinal cohort study that actually showed that if you have these ApoE2 or E4, you're at high risk of having hemorrhagic stroke over time. This is the trial from Progress trial looked at. It was actually a hypertensive trial that looked at over 5,000 people, Asians and Europeans, and then they knew their ApoE status and followed over time. Over time only means like 4 to 5 years. And they showed that if you have ApoE2 or 4, that you're at high risk of having this hemorrhagic stroke. And that was a lot more prominent amongst the Asian population compared to the Europeans. Just to blow it up, this is what it looks like amongst the Asians. So if you have ApoE2 or 4 compared to E3, then you have a higher risk of having a hemorrhagic stroke. Now, the x-axis only goes for like 4 years, so there's really no data. Like what goes on if you live, if this goes on for like 30 plus years, there's nothing like that in the literature. So, based on the Park program data, which has more than 34 years of data, then we can actually make a figure like that, and the x-axis would be 34, 35 years, and see what this graph would look like. Does it plateau out? Does it keep its widening? So that's our first research question, is to establish that. And then the second question is more of an exploratory question. So we know that there's some kind of a relationship between ApoE2, 4, and 3. So what happens if you have FOXO 3? Does it sort of buffer it out? Does it kind of look like the middle figure where if you have FOXO 3, even if you have a bad ApoE, it's going to sort of be a blanche? So what happens? Does it have a buffering effect? Again, it's an exploratory study. Like how does it sort of intertwine with each other? And the third question is, again, it's more exploratory, but, I don't know much about FOXO3 yet, but I perceive that as sort of a resilient gene. So if you have it, you're resilient. So maybe it's not going to change the incidence of having a stroke, but if you do have a stroke, you have a higher chance of bouncing back to normal life. So this whole hypothesis is if you don't have the longevity associated FOXO3 after a stroke, you're more likely to sort of dwindle and die earlier. Whereas if you have that longevity FOXO3 gene, if you have a stroke, you can hopefully bounce back and live much longer. So it's sort of what's the lifespan or longevity after the stroke if you have FOXO3? That's the third question that we're going to answer. Thank you.
Thanks for some very good presentations. And now we have the floor open for questions. Before I open it up though, I did want to acknowledge the Kuakini Honolulu Heart Program staff. Please wave your hands. Some of these people, some of them have been with the study from the early 80s. I don't even know how long ago that was, but it's a long time ago. And we also have some folks from Kuakini administration, so I wanted to welcome them too and thank them for their support. And now, does anyone have any questions? Any questions? Dr. Alsa.
Can I ask a question? This is actually a quick question for Kevin, actually.
Can you talk into the mic?
ApoE2 allele.
No.
When it's on? Yeah. Oh, maybe it's not on. There.
Oh, that's what. Yeah. So the ApoE2 allele, as you showed, it seems to have, and E4 seems to confer risk for hemorrhagic stroke, but the ApoE2 allele has been published is also protective against Alzheimer's. So can you kind of reconcile those two ideas?
It's a very good question. I don't know the answer. I intentionally didn't include a slide, but there is a different pathway for E2 and E4 in terms of how that impacts the amyloid angiopathy incidence or risk. I mean, the lines, I don't know. I don't know why, but we acknowledge that E2 and E4 are very different for the amyloid angiopathy relationship. But from the epidemiology standpoint, they're both at risk for the vascular angiopathy related. But I don't know why it's protective. I have no idea. It's just my guess, but it may just sort of shift more from the brain tissue pathways to the blood vessel pathway. It may sort of protect them from brain tissue deposition. I don't know. That's just my guess.
Other questions? Oh, go to the floor.
Okay. Oh, yeah. Okay, it's working now. I have a question for Jesse. So, assuming your mouse experiment works, and these mice that get the longevity version of the FOXO3 gene, and they live longer, healthier lives, what about humans? Will this become available for humans?
Jesse, if you would mind.
Yeah, so if that does work, there's a couple of ways that this could work in humans. You could either use a transposon technology like I'm developing to bring in another copy of FOXO, which is exactly what I'm doing. But the other thing that might work is, I didn't mention, is that you can introduce mutations into FOXO which make it more active. And the reason it's more active is because those mutations cause it to degrade slower. So if you introduce those mutations into a human, which would be a lot easier than bringing in a whole gene, we might be able to turn on more FOXO in people and get them to live longer, potentially. And there was a paper out that came out recently where they did this in a cardio cell line, and they showed that that cell line was resistant to the precursor steps to turning into cancer and more resistant to stressors. Yeah. So theoretically, we go in there and change some mutations and make FOXO stick around for longer, and you might live longer that way. Or you can just bring in a whole other FOXO gene.
Can I follow up with that? Just an idea. I was thinking, you didn't mention, I haven't thought about it or not, but what do you think about the possibility of using the CRISPR or genetic modifying technologies? Is it feasible to convert a T allele carrier who doesn't have a protective allele to having one or two copies of the gene by modifying the function?
Oh yeah. There's a new thing that just came out called base. There's base editing, which is a style of CRISPRs where it doesn't cut the genome, it just changes base pairs. That's one way you do it. And the other one's called prime editing, that's right. And so the prime editing one just came out a month ago, and that's like a new way of doing this where, again, you're just nicking the genome, you're not cutting it and introducing things. And they think that the efficiencies for that are pretty good. So maybe, the one thing you have to keep in mind is, and people always call me out on this, is that for that to work, you would have to get it into every single cell of the body. And that's not something that I work on, right? So my job is, once it's in the cell, send it somewhere safe. That's like my whole thing, right? So, but there is a whole field of people working on this, and how do we get systemic delivery? How do we get the thing that I'm making into all of your cells? And that's a much more mechanical engineering style of working, because they've got to figure out how to get through those cell walls and get your stuff in there. So yeah. Yes, that is the answer.
I seem to recall that there's some kind of moratorium or limitation on gene modification.
No, that's for babies. So the idea is that there's CRISPR therapies out now. So the problem with CRISPRs is they still have problems. When they cut the gene, whenever you cut the genome, you have a risk of a large deletion forming, and there's an additional risk of that chromosome sticking onto another chromosome somewhere else, which is called translocation, and that can cause cancer. So people aren't just using CRISPRs for all kinds of stuff, but they are beginning to use them for certain diseases where you're going to certain very dangerous diseases. And that's all okay because we're not messing with the germline. So if you have a therapy and then you have children, your children will not be modified. What is the moratorium is that you can't modify your children before they're born, because if those children have children, then their children will be modified, and that's not allowed. So we only do gene therapy on people in their lifetime and not mess with their sperm and eggs, so that their children will not be different because of our therapy. That's the rule. And I think that's the CRISPR baby thing that came out in China where they broke that thing. There are a lot of people doing that. so we don't do that. That's not allowed yet. Maybe someday when it's really safe, who knows? Maybe they might do that, but people are not doing that right now in normal labs. Oh, thanks.
Any other questions?
Thank you very much for our team of presenters for a very excellent and interesting presentation.